Welcome to Microscopic Marvels.
All right, today we're tackling a true heavyweight in the world of microbes, Staphylococcus aureus. You've probably heard of it by its more infamous name, the golden superbug. In this explainer, we're going to break down exactly what makes this bacterium one of the most important and dangerous pathogens we deal with in medicine.
So what is this thing really? Well, under a microscope, it's a gram-positive coccus, so it stains a deep purple. But the dead giveaway is its arrangement. They look like little grape-like clusters. It's also a facultative anaerobe, which is just a fancy way of saying it's tough. It can live with or without oxygen. And that name, Aureus, that's Latin for golden. And it's because when you grow it in the lab, it forms these beautiful golden yellow colonies.
Okay, here's our game plan. We'll start with its basic features, then dive into its incredible arsenal of virulence factors. After that, we'll look at the infections it causes, from simple skin stuff to sepsis. Then we'll see how we actually pin it down in the lab. And finally, we'll tackle the huge topic of MRSA and antibiotic resistance.
So, how does one tiny, single-celled organism cause so much chaos? It really all comes down to its arsenal of virulence factors. You should think of these as its biological toolkit, a set of weapons it uses to attack us, hide from our immune system, and spread throughout the body. We can pretty much group these weapons into three main categories. First, you've got its cell wall components, which act like armor and grappling hooks. Second, it pumps out a whole range of nasty toxins that directly damage our cells. And third, it releases enzymes that basically act like molecular scissors, letting it slice through our tissues and invade deeper.
Let's zoom in on one of its cleverest defenses, protein A. Now, you're going to want to really pay attention here because this is a super high-yield topic for exams. Here's the trick. Protein A sits on the bacteria's surface and grabs onto our own antibodies. But, and this is the genius part, it grabs them backwards by the Fc region. By doing this, it completely messes up the antibody's function. It's like it puts on an invisibility cloak made from our own immune system, preventing our killer cells from ever finding it.
And this isn't some rare feature either. Over 90% of the S. aureus strains that infect people have protein A. That number alone should tell you just how critical this one protein is to its success as a pathogen.
Now for the toxins. This table right here is your best friend for quick revision. Just link the toxin to the disease. Enter a toxin, that's your rapid onset food poisoning. TSST1, that's the superantigen that causes toxic shock syndrome. Epidermolytic toxin, think scalded skin syndrome. And PVL toxin, that's the one that shreds white blood cells, and you see it a lot in nasty community-acquired MRSA.
If you only have space in your brain for one enzyme from this talk, make it this one, coagulase. It is probably the most important weapon Staph has. It literally takes a component from our own blood, fibrinogen, and uses it to build a fibrin clot around itself. This forms a protective shield, hiding the bacteria from our immune cells and letting it set up shop, which is why Staph is so good at forming abscesses.
So with a weapons cache like that, it's really no surprise that S. aureus can cause a massive range of diseases, from a tiny pimple all the way to deadly body-wide sepsis.
Yeah, Staph is most famous for its skin and soft tissue infections. It can start simple, like folliculitis, but if it goes deeper, you get a boil or a furuncle, and if a bunch of those join together, you get a carbuncle. You'll see those a lot in diabetic patients. It also causes impetigo, which has that classic honey-colored crust appearance. And definitely remember this for surgery, S. aureus is the #1 cause of infections at surgical sites.
Okay, let's talk about that food poisoning. Here's the key takeaway, and it's a classic exam question. The incubation period is incredibly short. Why? Because you're not eating the bacteria and waiting for it to grow. You're eating a preformed toxin. That means within just 1 to 6 hours, you're hit with really intense vomiting. The good news? It's usually over in about a day. That super fast onset is your clue that it's staph.
All right, so you suspect you're dealing with a staph infection. How do you actually prove it in the lab? Let's walk through the detective work.
The workflow is very logical. First, you get a sample. Pus from an abscess, a blood sample, you name it. Then you gram stain it, looking for those classic gram-positive cocci in grape-like clusters. Next, you grow it on a culture plate, often using something selective like mannitol salt agar. And finally, you run a couple of key biochemical tests to be absolutely sure.
Okay, get ready. Here is a classic Viva question that you are almost guaranteed to be asked at some point. Both staph and strep are gram-positive cocci. So, in the lab, how do you tell them apart?
The answer is one simple test.
The catalase test. It's fast, easy, and definitive. You just add a drop of hydrogen peroxide. If it bubbles, it's catalase positive, and you've got Staphylococcus. If nothing happens, it's catalase negative, and you've got Streptococcus. You absolutely have to know this. Staph is positive, strep is negative.
So, we know it's staph, but how do we know it's the bad one, S. aureus? For that, we turn to the coagulase test. Now, there are two versions. The slide test is a quick screen, but the gold standard is the tube test. It detects free coagulase, and a positive result is unmistakable. You'll see a solid clot form in the test tube. Once you see that clot, you've nailed your culprit. It's S. aureus.
And now we get to the really hard part, fighting this thing. You see, the story of S. aureus is completely intertwined with the story of antibiotic resistance. And that leads us, of course, to MRSA.
So, what exactly is MRSA? It's right in the name, methicillin-resistant S. aureus. And the resistance comes from a specific gene called the MECA gene. This gene tells the bacterium how to build a modified protein, PBP2A. Now, our beta-lactam antibiotics, like methicillin, are supposed to attack this protein, but they can't bind to the new altered version. It's like the bacteria changed the locks, and our antibiotic keys just don't work anymore.
It's also super important to know that not all MRSA is the same. We split it into two main types, community-associated, or CA-MRSA, and hospital-associated, or HA-MRSA. CA-MRSA is the kind that causes skin infections in otherwise healthy people. HA-MRSA is the one we really worry about in the hospital. It tends to cause more severe, invasive disease, and it's often resistant to a whole bunch of different antibiotics.
This ladder of resistance basically tells us what drug to use. For a regular, sensitive staph infection, an MSSA, we can use drugs like cloxacillin. Simple. But if it's MRSA, those are useless. We have to step up to our drug of choice, vancomycin. And now, the truly alarming part. We're starting to see vancomycin-resistant staph, or VRSA. For those infections, we're down to our last line of defense with drugs like linezolid.
And this really leaves us with a critical and honestly a pretty scary question. We've watched this bug evolve past penicillin, then past methicillin, and now it's starting to beat vancomycin. As VRSA becomes more common, what's our next move? This really highlights just how urgent the threat of antibiotic resistance is and why we have to be so careful to protect a few effective drugs we still have.
If you found this explainer helpful, then please like, comment, and subscribe.